All right, so before I get started, I just want to thank the organizers for allowing me to give this talk today. This is my first big talk, so I'm happy to share it with you. I've also simplified my title, and I'm going to be talking more broadly about our lab's approaches to studying soil community response to warming. So in 2013, the Intergovernmental Panel on Climate Change established a series of projections for what the climate might look like over the next 100 years. And so they've projected atmospheric temperature change between a range of 2.6 and 8.5 degrees Celsius, with a 4-degree surface temperature change, which could have major implications for the health and function of our ecosystems. So the long-term ecological research site at Harvard Forest was established in central Massachusetts, and Dr. Jerry Melillo in 1991 had the foresight to establish the Prospect Hill warming plots, which are the longest-running warming experiment that I know of. And they were set up to establish the effect of a 5-degree temperature increase in soil on carbon fluxes. And in 2003, Jerry Melillo also established the Berry Woods warming experiment, which had larger plots and incorporated plant responses into this, our understanding of soil temperature increase on ecosystems. And in 2006, Sarita Fry from the University of New Hampshire established these soil warming and nitrogen plots, which look at the combined effect of nitrogen and soil warming on our ecosystems. And all three of these plots are ongoing to this day, so I'm going to talk about some of the results from them. And how these plots work, like I said, the heated plots are warmed 5 degrees above ambient temperature, and they have a series of underground heating cables which are buried at 10 centimeters below the soil surface. The control plots do not contain any heating cables, but they do have a temperature logger, and so this maintains the 5-degree temperature differential at all times. And what we're really interested in here is measuring the difference or delta between carbon dioxide flux between the heated and controlled plots and understanding the drivers of this difference in flux. So here, like I said, the Prospect Hill warming plot has been warmed for 25 years now. And we have taken uniform measurements of respiration over that past 25 years. And so here we have the delta respiration, so the difference between heated respiration and control, plotted over that first 10 years of warming. And so what we see immediately is this increase in respiration with warming treatment, where the respiration level is a lot higher in the heated plots as compared to the control. So that was a little terrifying. But as we move into phase 2, what we consider phase 2, in 2001, we see this attenuation response where heated respiration dips down to nearly control levels. So we started to relax a little. Maybe climate change isn't such a big concern in soil ecosystems. But in 2008, we again see this respiration flux that in the heated plots is a lot higher than in the control. And so our goal is to understand why we see this 3-phase pattern in respiration and try to understand why after this attenuation there's this increase in respiration in the heated plots once again. So our objectives are to understand soil community structure and function in response to long-term warming, to understand how warming might influence the active microbial community, and also how warming might alter the physiology of these active organisms. So in 2011, this group of PIs, including my advisor Jeff Blanchard, applied for the annual CSP call at the JGI, and were able to collect 48 soil samples across these 3 warming sites and establish a phylotype data set, which was published in 2015, a metagenome data set, which was published in 2016, and a Metatranscriptome data set, which is what our lab has taken a lead on and is what I'm going to show you guys today. So our first slide here is a comparison of the metagenome and metatranscriptome data sets. So we see the percentage of nucleic acids that are annotated to bacteria and eukaryotes on the y-axis. So when we look at the DNA perspective in the metagenome, we see that 98% of the reads are annotated to bacteria and less than 2% are annotated to eukaryotes. When we look at the metatranscriptome data, in contrast, we see 75% annotate to bacteria and approximately 25% are classified as eukaryotes. And so overall, we see a more holistic food web type overview of the ecosystem when we use this metatranscriptome approach. When we dig a little bit deeper and we look at the effect of warming treatment on the bacteria versus eukaryotes, we do see an increase in bacteria as compared to eukaryotes. So here we have our 3 warming experiments on the x-axis, and on the y is our bacteria to eukaryote ratio. And blue here represents the control treatment, and magenta is the heated. And what we're seeing is an increase with the heated treatment in bacteria relative to eukaryotes. And this includes acidobacteria, proteobacteria, and actinobacteria as our dominant groups, and they're increasing. And eukaryotes, including especially ascomycetes, are decreasing in response to warming. And so overall, eukaryotes are maybe responding more negatively to increased temperature. Additionally, when we annotate our mRNA to functional categories using seed, we experience a whole host of changes across biogeochemical cycling categories. And so I'm going to run through a few of those categories with you. First of all, in terms of carbon cycling, we see an increase in chitin utilization in the heated plots. So red shows an increase in heated categories, and blue shows an increase in the control plots. So we see an increase in chitin utilization in the heated plots and an increase in central carbon metabolism in the control plots. When we look at nitrogen cycling, we see an increase in nitrate and nitrite ammonification in the control plots. When we look at sulfur cycling, we see sulfur oxidation and alkane sulfonate assimilation increasing in the heated plots, which could indicate that there is a degree of sulfur limitation or stress in this environment. And when we look at the iron cycle, we see increases in iron acquisition and transport in the heated plots as well. So additionally, also potentially a indicator of iron stress or limitation. So although metatranscriptomics and metagenomics have provided great and valuable insights into our soil communities, there are still major challenges in studying this ecosystem. So anyone who's familiar with soil knows that there are estimates of up to 10 to the 9th cells of bacteria or fungi in every gram of soil. In this same gram of soil, there could be tens of thousands of microbial species, and less than 1% of this diversity has been isolated and sequenced. So we really know very little about the soil still. And what I really want to focus on in my research is the abundance of relic DNA that exists in the soil, and that comes from dead organisms and exogenous DNA, the lack of reference genomes that currently exist for the soil ecosystem, and this widely variant estimate of active organisms, which, according to a few papers I read, is between 10 and 50% of the total community. So my research is aimed at isolating or targeting these metabolically active organisms and finding ways to dig deeper into the dark matter that still exists in soil. So some of our ongoing studies, I just wanted to show how we are leveraging some of the resources available through JGI, although I don't have a lot of data to show today. So we're going to be doing deuterium labeling and Raman sorting in collaboration with the University of Vienna and the JGI, proteomics and deuterium, or lipid profiling of deuterium-labeled cells with EMSL through a ficus collaboration, and I'm currently working to develop BONGCAT, which is bioorthogonal non-canonical amino acid tagging, here at the JGI during my fellowship. So I'm mostly going to focus on this first point because this is what I have the most experience with right now. So this project was initially established through an ETOP collaboration with Miki Wagner's group at the University of Vienna. And we have stepped in with our ficus collaboration to work with them to develop this in our soil systems. So in December of this past year, I had the opportunity to take some samples with me over to Vienna that I had incubated with deuterium oxide, which is a heavy isotope of water. I extracted the cells using a Nikodin's gradient fractionation technique and sorted the label cells using a Raman-aided microfluidic device, which was developed at the University of Zurich. And we hope to apply a mini metagenomic sequencing approach to these sorted cells, which will give us nearly single cell resolution and provide us with the opportunity to construct some genomes from these organisms that have not been previously sequenced. So overall, we saw that 25% of our cells were labeled with deuterium, and this is very preliminary right now, which this falls in that 10 to 50% of the total soil community that we consider active. And so here I have an unlabeled spectrum and a labeled spectrum, and what we were looking for in this, these are for individual cells, we were looking for this peak at 2200 reciprocal centimeters, and so 25% of our total cells that we screened contained this peak, and we considered them metabolically active, and then in the future we would sort out these cells and sequence them using this approach. So we hope to analyze this data in the context of lipid profiling in conjunction with EMSL to gain some insights into our active soil community. So in summary, our metagenomic and metatranscriptomic approaches do reveal differences in community structure and function with long-term warming, although there are different views that we see using these two techniques. And we believe that function-driven genomics will provide new insights into the structure and function of the active microbial community and help us to understand these ecosystems. So I'd like to acknowledge my lab, especially William Rodriguez, who worked really hard on the CSP analysis, and Tanya for accepting me into her lab for this year. I really appreciate that. So thank you all for listening.
All right, we have time for a couple of questions. Yeah, Igor.
Great talk and interesting results so far. When you say active, what season did you sample? Because I imagine the active community changes throughout the year.
Yes. So this was one time point collected in October of 2011. So right now we're currently working on a time series. So we will have samples collected between April and October, and we should have an idea, a better idea of the active community using metatranscriptomics that way.
So you mentioned chitin utilization increase. Does it mean chitin production or deconstruction? So chitin is present in fungi.
Right.
So does it correlate with fungal growth or opposite?
We see chitin utilization increasing, so that is the breakdown of chitin. And because we're seeing this decrease in fungi and an increase in bacteria, we do think that that is related to bacterial metabolism of chitin. And we actually see when we use KZ to annotate our data, we see an increase in CBM2, which is bacterial carbohydrate utilization. So overall, I think there's an increase in that bacterial role in the ecosystem.
So I'm just kind of wondering about the control, or well, not just the control, but the field plots. So you have the ones that do not have any heat, and then you have the heated ones. But what about other factors? I mean, are they covered? I mean, how much water are they getting? And I'm just kind of wondering, like, are you able to monitor or isolate other factors that could be influencing your metagenomic community structure?
So these are adjacent to one another in the fields. They don't have any other, like no rain exclusion or anything like that. So they roughly receive the same amount of moisture, and we do keep track of soil moisture, like temperature, at all times. But there's a lot of variability that we can't always control for, so that makes this more difficult, definitely.
All right. So did we have, I think, maybe we'll take one more here, and then we'll.
Thank you. Interesting that in your metagenomic and metatranscriptomic data, just no reads that related to archaea?
We have, it's like less than 1% of the total community, but we do see some archaea.
How much do you think that might be influenced on references? I always wonder, like, what percent of reads perhaps are going unassigned, and is it in part due to poor reference problems? But it just struck me that that there might be some archaea there based on what we know.
Right. Yeah, it very well could be a reference issue. We tried our best not to, as everyone does,
I
not to.
understand,
All right. Well, thanks so much to all the speakers. The lunch is going to be, I think, up the stairs. There should be people and signs guiding you when you head out of the room. And let's give all the speakers a hand for such a great session.